The plasma membrane is an important component of the cell. It will help to enclose the cell and contain all of the different organelles and components. It is important in functioning as a selective barrier, which means that it will let certain molecules in as well as waste material out of the cell. It will also have various types of protein channels that will help get substances in and out of the cell, such as aquaporins that will specifically let water molecules to pass across the membrane. The plasma membrane will also contain other types of proteins, also glycoproteins and glycolipids, which are simply proteins and lipids with sugar chains attached to them, as well as various cholesterol molecules inserted in the membrane as well. This will help to allow for different components to float or move throughout the plasma membrane. The nucleus is an important structure of a eukaryotic cell. It is surrounded by a lipid bilayer called the nuclear envelope. This nuclear envelope has large pores that will allow for specific molecules to enter and exit the nucleus. The nucleus will house the DNA, which is usually in the form of chromatin or unwound chromosomes. This is important to understand. When a cell is functioning, it needs to constantly make proteins, which means that the DNA is constantly being transcribed and eventually translated into those proteins. As a result, the DNA needs to be in a readable form, which would be in the form of chromatin. DNA is only wound up in the form of chromosomes when a cell needs to divide. The nucleus also contains what is termed the nucleolus. This is a dense region within the nucleus that is a site of ribosomal RNA transcription. This is the birthplace of ribosomes. Ribosomes are responsible for making all of the proteins in the cell. If you take a look at their base structure, they're comprised of two subunits, one large and one small. However, both of these subunits are going to be made of ribosomal RNA produced in the nucleolus, as well as protein. There are two main types of ribosomes present in the cell. Free ribosomes are termed free because they are free-floating in the cytosol. These are responsible for making proteins that are located in the cytosol, in the nucleus, mitochondria, etc. Some of these ribosomes will be bound to the rough ER. These ribosomes are responsible for making proteins located in the ER, Golgi apparatus, plasma membrane, lysosome, or any proteins that are going to be secreted outside of the cell. It is important to note that all of the bound ribosomes initially start as free-floating ribosomes. However, there will be a signal on that polypeptide chain that designates it as a specialized protein that needs to be produced in the ER. As a result, that ribosome will be directed to the ER and then bound to it to carry out protein synthesis. It is important to describe the concept of the endomembrane system. The endomembrane system consists of a network of internal membranes within the cell. These membranes have a similar structure as the plasma membrane, which means that they will be comprised of a lipid bilayer. There are major components of the endomembrane system that consist of the nuclear envelope that extends into the ER, smooth and rough, followed by the Golgi apparatus, and then lysosomes, vacuoles, and eventually the plasma membrane. The endomembrane system is important for protein trafficking. This is because material or proteins will be exchanged from one compartment to the next, and this material is going to be moved within membrane-bound vesicles. So as a protein is produced in the ER, and when it needs to travel to the Golgi, it is going to be packaged in a vesicle delivered to the Golgi apparatus. The endoplasmic reticulum is a very extensive network of membranes that lie directly outside of the nucleus. It comprises more than half of the total membrane of some eukaryotic cells. It is considered to be continuous with the nuclear envelope, meaning that there will be extensions throughout the nuclear envelope that will extend into the endoplasmic reticulum. The inside of this endoplasmic reticulum can be termed the lumen. There are two different types of endoplasmic reticulum, rough and smooth. They differ with respect to structure and function. The rough ER is covered with ribosomes. Some of the proteins that are going to be synthesized or injected into the rough ER are going to be created by ribosomes that are bound to the ER. This means that all of the proteins that are destined for the plasma membrane or destined to be secreted from the cell, or anywhere that are going to be used in the endomembrane system, either in the Golgi or lysosomes, etc., are all going to be produced by the rough ER. The rough ER is considered to be the first stop shipping center for many proteins. The smooth ER does not have ribosomes attached to it. However, it performs very different functions. The smooth ER is responsible for synthesizing lipids, steroid hormones, and as well as carbohydrates. It's responsible for the detoxification or neutralization of various drugs and poisons, as well as calcium storage, which is particularly important in muscle cells. This is because muscle cells need a high level of calcium in order to be able to contract their filaments properly. This image here depicts the difference between the rough and smooth ER. Note that the ribosomes are present on the rough ER. Also note that the endoplasmic reticulum exists as small extensions from the nuclear envelope. So you will see in this image little small attachment sites between the nuclear envelope and the ER. The next organelle we will discuss is the Golgi apparatus. The Golgi apparatus consists of a stack of membranes or cisternae. There are two sides to the Golgi. One side that faces the ER, we call this the cis side, and it will be receiving vesicles that are delivered from the rough endoplasmic reticulum. There's also the trans side, and this would be the side facing the plasma membrane. This is the area where vesicles will leave the Golgi apparatus and enter into the cytosol to then be delivered to their final destination. The Golgi apparatus is considered to be the second stop in the shipping center for many proteins. In the Golgi apparatus, most proteins are further modified, meaning that they're fully folded and matured, and they may also undergo the process of glycosylation. This means that if you have a glycoprotein that needs to have a carbohydrate chain attached to it, this would occur in the Golgi apparatus. Once the protein is fully mature and modified, it will then be sorted and shipped to its final destination, as in other parts of the endomembrane system, such as the plasma membrane or lysosome or peroxisome. Let's take a look at the structure of the Golgi apparatus. We can see that the Golgi apparatus lays in close association with the endoplasmic reticulum. Specifically, the cis side would be considered the receiving side and be the side closest to the ER. We can see that there are many tube-like cisternae structures that are stacked upon each other. We then refer to the trans side as the side that is facing the plasma membrane. As vesicles are received into the cis side, they will travel through the Golgi apparatus. Those proteins will be further matured and modified, glycosylated if need be, and then packaged into vesicles on the trans side and delivered to their final destination. The lysosome is also an important organelle of eukaryotic cells. This is considered to be the stomach of the cell. It's responsible for digesting food from phagocytosis and also for undergoing autophagy, meaning that it will help digest any organelles or proteins within the cell to be recycled. The lysosome structure is a membrane-bound sac that contains many hydrolytic enzymes for digesting or breaking down those macromolecules that we learned about in the previous lecture. Remember that in order to form those macromolecules, we have to undergo dehydration synthesis, and in order to break apart those macromolecules, we would undergo rehydration or adding water. So these hydrolytic enzymes will be adding water to help break apart those macromolecules. This is an example of a cell ingesting a food particle. In this instance, you would have a pseudopod that would go around and then bring this molecule into this food particle into the cell. You can also see the darkened circle areas, which would be the lysosomes. So as this food particle is brought into the cell, the lysosomes would then fuse to the membrane, release their hydrolytic enzymes, and start digesting it. An important thing to know about the structure of lysosomes is that it has a very acidic environment, very much like the stomach. Lysosomes sit at about a pH of 5. That's important to note because the overall pH of the interior of the cytosol sits at about 7.4. Because it is acidic, this will help to break things down into monomers. What is also important is that the hydrolytic enzymes that are inside of the lysosome will only work at a low pH. This is important because if a lysosome were to break and these hydrolytic enzymes were released into the cytosol, it would be detrimental to the cell if they were active at a cellular pH. So by having them contained in a lysosome and by having them only active at a low pH, this will help to maintain the integrity of the cell. Remember that the lysosome will be important in fusing to these vesicles. Whether these vesicles are formed from ingesting molecules or whether these vesicles hold very important organelles that are going to be recycled, the action of the lysosome remains the same. The lysosome will bind to and fuse to the vesicle. It will release the hydrolytic enzymes to the interior, which will then help to digest the material. Vacuoles are another important structure of this eukaryotic cell. Vacuoles are membrane-bound sacs that can have very different functions depending on the cell type. Some vacuoles can be termed food vacuoles when they result from phagocytosis, and they can be used for the storage of food particles. Some vacuoles can be contractile by pumping excess water out of the cell. And in plant cells, they have a very important central vacuole, which is a very large vacuole that can hold a variety of substances, most importantly water. This will help to maintain the osmotic pressure of the cell. The central vacuole can also contain ions, proteins, different toxins, defense chemicals, and the central vacuole overall, the membrane, is a bit tougher in the central vacuole, and we can refer to that as a tonoplast. So let's just review the concept of the endomembrane system. All proteins that are destined for any part of the endomembrane system, remember, are going to be synthesized by ribosomes on the rough ER. They will either be injected into the ER if they are going to be an internal type protein or a secreted protein, or they will be injected into the actual membrane itself if they are going to be a glycoprotein, a transmembrane protein, etc. Once these proteins are produced in the ER, they will travel to the Golgi apparatus where they will receive some sort of modification, and there is where they will mature. At the trans side of the Golgi apparatus, they are going to be sorted out and then be delivered in vesicles to their final destination. This slide here reviews the components and the trafficking within the endomembrane system. Remember, we have the nuclear envelope that extends into the rough and smooth ER. Any protein that is going to be secreted or part of the plasma membrane or part of a lysosome or any other part of the endomembrane system is going to be made by the rough ER. It's going to be packaged in vesicles delivered to the cis side of the Golgi. It will then be matured, glycosylated if necessary, and then it will exit the Golgi through the trans side. From there, it will be delivered to a lysosome if that's its final destination, if it's a hydrolytic enzyme, or it could be delivered to the plasma membrane where it will either be secreted if it is an internal protein, or it will be inserted into the plasma membrane if it is an embedded protein.